know, a major part of modern instrument. Hello students. In this lecture, we are going to discuss different non-classical methods of chemical analysis based on optical properties of the sample. In the beginning, let us see the introduction about optical methods. As we know, a major part of modern instrumental analytical chemistry focuses on the study of energy exchange between electromagnetic radiations and matter. These interactions are visible to the naked eye when the radiations concerned fall within the visible spectrum. So what are optical methods? Optical methods are based on how the sample acts towards electromagnetic radiation. The absorption or emission of radiant energy, the bending of radiant energy, the scattering of radiant energy, and the delayed emission of radiant energy are typical optical properties which can be correlated to the concentration of the sample. Optical methods have extensively applied to distinguish various materials whose chemical composition and crystalline structure exhibit a particular behavior when interacting with the full light spectrum. So herein we are going to discuss some important instrumental techniques based on optical properties, namely flame emission spectroscopy, which is called as flame photometry. Second, atomic absorption spectroscopy. These two are the examples of atomic spectroscopy. Then fluorimetry and phosphorometry, which is an example of molecular spectroscopy. And last, turbidimetry and nephelometry. So these four instrumental techniques we are going to study in today's lecture. Let us see about the first important technique, that is flame photometry. So flame photometry is an example of atomic spectroscopy. So what do you mean by atomic spectrum? The range of characteristic frequencies of electromagnetic radiation that are readily absorbed and emitted by an atom. When these atoms are excited, they emit light of certain wavelengths which corresponds to different colors. The emitted light can be observed as a series of colored lines with dark spaces in between. This series of colored lines is called a line or atomic spectra. So each element produces a unique set of spectral lines. That's a special identity of each element. These are the emission lines of the different atoms, specifically sodium, hydrogen, calcium, magnesium, neon. Each element's emission spectrum is distinct because each element has a different set of electron energy levels. That's why each element shows different emission spectrum. Let us see the basic principle of flame photometry. When a solution of any metallic salt is sprayed or sprinkled as fine droplets into the flame, due to heat of the flame, the droplets dry, leaving a fine residue of salt. This fine residue converts into neutral atoms. Due to the thermal energy of the flame, these neutral atoms get excited and thereafter return to ground state. In this process of return to ground state, these excited atoms emit radiation of a specific wavelength. This wavelength of radiation emitted is specific for every element. Let us understand the principle of flame photometry with the help of this schematic diagram. When a metal salt solution is aspirated into the flame in the form of aerosol, the following events takes place in rapid succession. The solvent evaporates, leaving behind tiny solid particles of the salt. These solid particles vaporize into gaseous molecules, which are then converted into gaseous atoms by thermal dissociation process. Some of the gaseous atoms absorb thermal energy or heat energy from the flame and are excited to higher energy state. The fraction of metal atoms which are excited is calculated by Bole's distribution law. As we know, the excited atoms are unstable and short-lived. They emit visible light or sometimes UV light and return to ground state. The intensity of the emitted light is directly proportional to the excited atom population, and the measurement of the intensity of the emitted radiation permits a quantitative estimation of the element, and the determination of the wavelength of emitted radiation helps us to identify the element present in the sample. This diagram explains the phenomena of absorption and emission of radiation. So initially, atoms or gaseous atoms absorbs the energy from the flame and goes to excited state or higher energy state. As this excited state is unstable, so they again return back to ground state by emitting the amount of energy absorbed. In this process, the wavelength of light gives us an idea about the element present in the sample. That is a qualitative estimation. And the color's intensity tells us how much of the element concentration present in the sample. That is a quantitative estimation. So in conclusion, wavelength of light emitted gives us idea about the element present in the sample, and color intensity gives us idea about the concentration of the sample. That is, this method can be employed for both qualitative as well as quantitative analysis. Under constant and controlled conditions, the light intensity of characteristic wavelength produced by each of the atom is directly proportional to the number of atoms that are emitting energy, which in turn is directly proportional to the concentration of the substance of interest in the sample. Various metals emit a characteristic color of light when heated. For example, sodium emits yellow light, potassium emits violet light, barium emits green light, calcium emits orange light, and lithium emits red light. So these characteristic colors gives us an idea about the element present in the sample. These characteristic flame colors represents the presence of particular element in the sample, like yellow light for sodium, violet light for potassium, orange light for calcium, red light for strontium or lithium, green light for barium or copper, and blue light for lead or arsenic, etc. A flame photometer has the following components. First, flame atomizer or burner, then nebulizer, filter, photodetector, and readout device. Let us discuss these components in detail one by one. Let us discuss about the flame atomizer or burner. A burner that provides flame and can be maintained in a constant form and at a constant temperature. The sample is usually introduced into the flame in the form of an aqueous solution. Substances which are not soluble in normal solvents are first decomposed and then dissolved to form solutions. For example, soil samples, animal and plant tissues, blood and petroleum products, etc. These substances may be decomposed by the treatment with hot mineral acids like sulfuric acid, nitric acid and perchloric acid. The second component is nebulizer and mixing chamber. After the appropriate treatment, the substances are dissolved in suitable solvents to obtain the sample solutions. The sample solution is first converted into fine droplets. This process of converting the sample into fine droplets is called nebulization process. The atomization device used in flame photometry consists of nebulizer. The nebulizer converts the sample solution into a fine spray or aerosol, and which is then introduced into the flame. Third important component is wavelength selector, that is filter or monochromator. If the reflected beam consists of a small number of wavelengths, an optical filter is used. This optical filter allows the radiation characteristic of the element to pass through. Whereas, if the reflected beam consists of a large number of wavelengths, a monochromator is used. It is used to disperse the reflected beam into its individual wavelengths. When the monochromator is rotated, the different wavelengths are focused one by one on the photocell or photodetector. This scanning enables us to detect several elements present in the sample simultaneously. Two types of monochromators are usually used in flame photometry are quartz or glass prism and diffraction gratings. A diffraction grating produces greater dispersion as compared to nickel prism. Then the next but most important component of flame photometer is photodetector. This photodetector detects the emitted light and measures the intensity of radiation emitted by the flame. That is, the emitted radiation is converted to an electrical signal with the help of photodetector. The produced electrical signals are directly proportional to the intensity of light. Photocell and photomultiplier tube are the commonly used detectors in flame photometry. Let us see the different types of burners or flame atomizers used in flame photometry. Generally, two types of burners are used in flame photometry. First, total consumption burner, and second, premix or laminar flow burner. This is the schematic diagram of total consumption burner. In this burner, fuel and oxidant are usually H2 and O2 gases. Sample solution is aspirated or sprinkled through a capillary with the help of high pressure of fuel and oxidant and burnt at the tip of the burner. In this type of burner, entire sample is consumed. Therefore, it is called as a total consumption burner. In a premix burner, only the small droplets of the sample
reach the flame, as the large droplets are trapped by baffles and drained off. Hence, only about 5% of the sample is introduced into the flame and the rest is wasted. But this small percentage is responsible for the efficient atomization of the sample. Due to the large wastage of sample, fewer atoms are excited and consequently the emission intensity is weak. And this lowers the sensitivity of the estimation. But the advantage of this laminar flow burner is the flame produced is stable, non-turbulent, noiseless and large in cross section.
Let us see different types of monochromators used in flame photometry. What is monochromator? A monochromator is an optical device that transmits a mechanically selectable narrow band of wavelengths of light or other radiation chosen from a wider range of wavelengths available at the input. There are two commonly used monochromators in flame photometry, that is Nicol prism and diffraction grating. Nicol prism is an optical device which is used for producing and analyzing plane polarized light. And like a prism, a diffraction grating separates the colors present in white light to produce a spectrum. If the reflected beam consists of a large number of wavelengths, a monochromator is generally used. It disperses the reflected beam into its individual wavelengths. Flame emission spectroscopy is mostly used for the detection of alkali metals and alkaline earth metals. A filter flame photometer is used for the detection of a small number of elements like sodium, potassium and calcium. However, using a monochromator, many elements can be detected simultaneously in the presence of one another. There are different methods used for the quantitative analysis of the samples by using flame photometer, of which first method is calibration curve method. In this calibration curve method, a series of standard solutions of known concentrations of the element is first prepared. Usually solutions of 5 ppm, 10 ppm, 15 ppm, 20 ppm, 25 ppm and 30 ppm along with unknown solution. These solutions are then introduced one by one into the flame. Using the proper filter, the intensity of emission is determined for each standard solutions. Similarly, the unknown solution is introduced into the flame and its emission intensity is also determined. Then after, a graph of intensity measured versus concentration is plotted. From this straight line graph passing through the origin, the concentration of the element in the unknown solution can be easily determined. For this, plot the emission intensity of unknown solution on the intensity axis and draw the line parallel to the x-axis where it cuts the standard curve. From there, draw the line perpendicular to the concentration axis, which will give the concentration of unknown solution. In this way, the calibration curve method is one of the common methods used for the quantitative estimation of unknown samples. Second method is standard addition method. In this method, the emission intensity of the test solution is measured with reference to the blank. The sample solution of unknown concentration X is aspirated into the flame and its emission intensity is determined with reference to the blank. Then, a series of solutions containing a definite amount of the unknown solution X and varying amounts of the standard solutions, that is 5 ppm, 10 ppm, 15 ppm, 20 ppm and 25 ppm, are aspirated one by one into the flame and their emission intensities are determined with reference to the blank. Then a graph of emission intensities versus standard concentration added is plotted. The graph will be straight line. This line is extrapolated on concentration axis, which gives the concentration of unknown solution. Let us see the applications of flame emission spectroscopy. The flame emission spectroscopy can be used to determine the availability of alkali and alkaline earth metals, which are critical for soil cultivation. In agriculture, the fertilizer requirement of the soil is analyzed by flame test analysis of the soil. Then another application is in the clinical field. Na plus and K plus ions in the body fluids, muscles and heart can be determined by diluting the blood serum and aspiration into the flame. The flame photometry can also be used for the analysis of soft drinks, fruit juices and alcoholic beverages. Let us see the advantages of flame emission spectroscopy. It is a simple quantitative analytical test based on the flame analysis. This method is an inexpensive method. The determination of elements such as alkali and alkaline earth metals is performed easily with this most reliable and convenient method. This method is quite quick, convenient and selective and sensitive to even parts per million, that is ppm, to parts per billion, that is ppb range. Here are some limitations of this flame emission spectroscopy. With the help of this method, limited number of elements that can be analyzed. Then the sample requires to be introduced as solution into fine droplets. Many metallic salts, soil, plant and other compounds are insoluble in common solvents. Hence, they can't be analyzed by this method. In case of volatile samples, if the small amount of sample is present, it is tough to analyze by this method as some of it gets wasted by vaporization. Then another limitation is alteration of light emission because of altered flame temperature. It needs perfect control of flame temperature. Interface by other elements is not easy to eliminate it, and in case of heavy and transition metals, the number of absorption and emission lines are more and the spectra is complex. These are the several limitations of this flame emission spectroscopy.
Hello friends.
Now
We are going to discuss about the basic principle, instrumentation and applications of atomic absorption spectroscopy. And we also see how this atomic absorption spectroscopy is more superior than flame emission spectroscopy. Let us see what is atomic absorption spectroscopy.
What is atomic absorption spectroscopy? Atomic absorption spectroscopy is the study of absorption of radiation by the ground state atoms when a sample is introduced into a flame. Of course, this radiation is the same as that emitted by the excited atoms of the element in the flame photometry. This absorption of characteristic radiation helps in the identification of the absorbing element. The intensity of absorption depends on the amount of sample introduced into the flame. Hence, atomic absorption spectroscopy is an important tool for the qualitative and quantitative estimations of a large number of elements. This important method was discovered by Sir Alan Walsh in 1955. In analytical chemistry, it is a very important technique for determining the concentration of a particular metal element in the sample. And it can be used to analyze the concentration of more than 62 different metals in the solution. That's why, as compared to flame emission spectroscopy, in application point of view, this atomic absorption spectroscopy is more superior. Let us see the principle of atomic absorption spectroscopy. So the principle of atomic absorption spectroscopy is again similar to that of flame emission spectroscopy to certain extent. As like flame emission spectroscopy, when a metal salt solution is introduced into a flame, the solvent evaporates, leaving behind the tiny solid particles of a salt. The solid particles vaporize into gaseous molecules, which are then converted into gaseous atoms. Most of the atoms remain in the ground state. These atoms are capable of absorbing their own characteristic radiation. If the light of this characteristic wavelength is allowed to pass through a flame containing these atoms, absorption takes place. The amount of radiation absorbed is proportional to the number of absorbing atoms present in the flame. And this absorption depends on the number of unexcited atoms present in the flame, and hence is dependent of the flame temperature. As against this, in flame emission spectroscopy, the emission intensity depends on the number of excited atoms and hence on the flame temperature. This is the basic difference in flame emission spectroscopy and atomic absorption spectroscopy. So in flame emission spectroscopy, flame temperature is responsible to subject ground state atoms to excited state, whereas in atomic absorption spectroscopy, the characteristic wavelength is responsible for the excitation of ground state atoms to excited state. This is the instrumentation of atomic absorption spectrophotometer. The components of an atomic absorption spectrophotometer are the radiation source, which is hollow cathode lamp, the rotating chopper, atomization unit, monochromator, detector, amplifier, and lastly, the readout device, or you may call it as a recorder. Up to this atomization unit, the instrumentation part is common as that of flame emission spectroscopy. whereas only the difference is here. So additional hollow cathode lamp is used in atomic absorption spectroscopy. Let us discuss one by one the components of this atomic absorption spectrophotometer. So first component is radiation source. In atomic absorption spectroscopy, hollow cathode lamp is used as a source of radiation. It consists of a hollow cathode cup made up of the element which is under study. For example, if copper is to be detected in the sample, the lamp will have a hollow copper cup as a cathode. The cathode is protected in a glass shield. A tungsten wire functions as an anode. The two electrodes are enclosed in a glass envelope containing an inert gas like neon or argon at a low pressure. So this is the construction of hollow cathode lamp. Let us see how this hollow cathode lamp functions. When a voltage of around 300 volts is applied between the two electrodes, the atoms of the inert gas ionize and the positively charged ions are attracted to the cathode. These fast moving positive ions strikes the surface of cathode, causes sputtering of metal ions or removal of metal ions from the surface of cathode. These metal ions now collide with the highly energetic gas ions and get excited. These excited metal atoms emit their characteristic radiation, which usually consists of a small number of individual wavelengths. This emitted radiation appears as a bright glow in the hollow cathode cup. Each hollow cathode lamp emits its characteristic wavelengths associated with the line in the spectrum of metal. For example, for the analysis of nickel in a sample, a nickel lamp is to be used. For the study of aluminum, an aluminum lamp is to be used. This somewhat restrict the number of elements which can be analyzed using atomic absorption technique. So this diagram explains this whole phenomena. These fast moving ions strikes over the surface of cathode and removes the surface metal atoms. This process is called as a sputtering process. Then after these metal atoms collides with these high energetic gas ions and get excited. And these excited metal atoms emit their characteristic radiation and return back to ground state. As we previously discussed, the type of hollow cathode tube depends on the metals being analyzed. Such as for analyzing the concentration of copper ions in a ore sample, a copper cathode tube would be used. And likewise, different hollow cathode lamps are required for different metals. A rotating chopper is placed in front of this hollow cathode lamp. This rotating wheel breaks the beam of light coming from hollow cathode lamp, and the resulting intermittent light produces a pulsating current in the photomultiplier tube. That is alternating current in the photomultiplier tube. This pulsating current is then amplified in an AC electronic amplifier.
After halocathode lamp and rotating chopper, the third important component of atomic absorption spectrophotometer is atomization unit. Two types of atomization units used in atomic absorption spectroscopy. First one, flame atomizer, and second one, electrothermal atomizer. As like in flame photometry, these flame atomizers or burners are of two types: total consumption burner and laminar flow burner. The construction and working of both these burners we already discussed in flame photometry. We just recap here about the flame atomizer. In case of flame atomizer, a flame is produced by burning a fuel gas like hydrogen in the presence into the flame and aspirated by oxygen to form solid particles. The solid particles vaporize and finally get converted into gaseous atoms. These gaseous atoms in the ground state absorb their characteristic wavelength from the beam coming from the hollow cathode lamp. As we know, there are several disadvantages of this flame atomizer. In case of laminar flow burner, the sensitivity of a flame atomizer is less, as a large amount of sample is drained off and hence not reached to the flame. And in case of total consumption burner, turbulent flame is produced due to the formation of larger droplets. These are the main drawbacks of these flame atomizers. So to overcome these drawbacks, this electrothermal atomizer can be used. This electrothermal atomizer is more sensitive as the entire sample is atomized and the time of stay of the atoms in the optical beam is about 1 second, which is more as compared to flame atomizers. In an electrothermal atomizer, the sample is first heated at a relatively low temperature to remove the solvent by the process of evaporation. It is then ashed at higher temperature on the electrically heated carbon surface. At the electrically heated carbon surface, which is in the shape of a hollow tube. After ashing, the temperature is increased rapidly to 2000 to 3000 degrees centigrade to atomize the sample in a few milliseconds. The absorption of radiation by the atomized sample can then be measured above the carbon surface. This electrothermal atomizer consists of a graphite tube open at both the ends. This tube is 5 cm long and has an internal diameter of 1 cm. The sample is introduced through a central window in the tube. Two electrodes are fitted at the end of the tube and a stream of an inert gas like argon or nitrogen flows in and around the tube, which is to prevent the possible oxidation of the graphite material. The entire assembly is then kept in a water-cooled jacket. It is popularly referred as a graphite furnace. So what are the advantages of this electrothermal atomizer? These are namely temperature that is enough to ash the sample and to produce the metal ions. The sample volume is very small. There is no need for fuel oxidant mixture. It is highly sensitive. No flame noise or turbulence. Solid samples can be used directly in electrothermal atomizer and heat distribution is uniform and temperature is steady. These are the several advantages of this electrothermal atomizer over flame atomizers. Then the next important component is monochromator. So the function of monochromator is same as that of in case of flame emission spectroscopy. Here also nickel prism or diffraction grating are used as a monochromators as like in flame emission spectroscopy. And the function of the monochromator is to select a given absorbing line from spectral lines by emitting hollow cathode lamp. The next component is photodetector. As like in flame emission spectroscopy, in atomic absorption spectroscopy also uses photocell or photomultiplier tube as photodetectors. These photodetectors detect the intensity of the radiation by converting the light energy into an electrical signal. The produced electrical signals are directly proportional to the intensity of the light. Let us see what are the important applications of atomic absorption spectrophotometry. This technique can be used for water analysis, that is the presence of calcium, magnesium, iron, silicon, aluminum or any other elements present in the different water samples can be easily analyzed with the help of this atomic absorption spectroscope. Then this technique can also be used for the analysis of foods. Various metal salts are used as a food preservatives, and the content of these food preservatives or metals in the food can be easily analyzed with the help of this atomic absorption spectroscopy. Then the third important application is analysis of animal feedstuffs. Animal feedstuffs also contains some food preservatives as well as some salts, and the metal content present in these animal feedstuffs like manganese, iron, copper, chromium, selenium, zinc, etc. can be analyzed with the help of this AAS spectroscopy. Then another application is analysis of additives in lubricating oils, greases, etc. One more application which is very much important, that is analysis of soil samples. Analysis of soil samples is very much important in farmers' point of view, because what type of crops to be cultivated in the farm is carried out on the basis of this soil sample analysis results. That is, what type of metals or what type of nutrients which are present in the soil, that can be analyzed with the help of atomic absorption spectroscopy. And the last but very important application of this atomic absorption spectrophotometer is in clinical analysis. That is, for the analysis of electrolytes like sodium, potassium, calcium, magnesium, lithium, etc. are present in the blood samples can be analyzed with the help of atomic absorption spectroscopy. These are various applications of atomic absorption spectroscopy. Let us see how this atomic absorption spectroscopy is more superior over flame emission spectroscopy. In case of A.S., solutions, slurries and solid samples can be easily analyzed. In atomic absorption spectroscopy, much more efficient atomization techniques can be used. A.S. has greater sensitivity over flame emission spectroscopy as there is a minimal loss of sample. Smaller quantities of samples are required. Typically 5 to 10 microliter of the sample is enough for atomic absorption spectroscopy, whereas very large quantity of sample is required for flame emission spectroscopy. And the last, A.S. provides a reducing environment for easily oxidized elements. So these are the important advantages of this atomic absorption spectrophotometer over flame photometer. That's why atomic absorption spectrophotometer is widely used over flame photometer. But there are certain disadvantages also over flame emission spectrophotometer. Visually, A.S. is very much expensive, which has low precision, low sample throughput, and it requires high level of operation skills. These are the important disadvantages of atomic absorption spectroscopy, as it is more expensive, low precision, low sample throughput, and requires skilled operator in comparison to flame emission spectroscopy. Let us see some distinguishing points between flame emission spectroscopy and atomic absorption spectroscopy. In case of A.S., solutions, slurries and solid samples can be analyzed, whereas in case of flame emission spectroscopy, Only samples in the form of solutions can be analyzed. Efficient atomization is possible in both AS and FES. AS has greater sensitivity, whereas flame photometry has less sensitivity. In case of AS, small quantities of samples, typically 5 to 50 microliter is enough, whereas large quantities of samples are required in flame photometry. Along with alkali metals, transition metals can also be determined with the help of atomic absorption spectrophotometer, whereas only alkali and alkaline earth metals can be determined with the help of flame photometry. In case of AS, skilled operator is required, whereas in flame emission spectroscopy, highly skilled operator is not required. So anyone can easily operate flame photometry, but skilled operator is required for atomic absorption spectroscopy. So these are some distinguishing points between atomic absorption spectroscopy and flame emission spectroscopy. This atomic absorption spectroscopy can be used for qualitative as well as quantitative analysis of metal samples. Let us see how these methods can be used for the quantitative analysis of metal samples. There are different methods are used for the quantitative analysis, which are calibration curve method, standard addition method, and internal standard method. Let us see in detail this calibration curve method. We already discussed this method in flame photometry. In this method, a series of standard solutions containing known amount of the element is prepared. For example, 1 ppm to 10 ppm. The standard solutions and the unknown are then introduced one by one into the flame, and their absorbance values are determined. Before each solution is sprayed into the flame, distilled water is introduced into the flame and the reading is adjusted to zero absorbance. This is more important step to remember. Once we record the absorbances of standard solutions and unknown solution, the absorbance values are plotted against the concentration of metal in the various standard solutions. And from this linear calibration curve, the concentration of the unknown solution can be easily found out. At the end, plot the absorbance of the unknown solution and then extrapolate this line parallel to concentration axis. The second method is standard addition method. As like in flame photometry, in this method also, the sample solution of unknown concentration X is aspirated into the flame and its absorbance is determined with reference to the blank. Then a series of solutions containing a definite amount of the solution X and varying amounts of the standard solution S, that is 1 ppm, 2 ppm, 3 ppm up to 10 ppm, are aspirated one by one into the flame and their absorbance values are determined with reference to blank. A graph of absorbance is plotted against the concentration of X and the concentration of S. The straight line graph obtained is extrapolated to intersect the concentration axis. The point of intersection gives the concentration of the unknown solution X.